Hello everyone, my name is Xin from the Department of Statistics in Florida State University. Today I'll be talking about text mining drug-protein interactions using an ensemble of BERT, sentence BERT, and T5 models. This project is done by me, Wanjing Wang, and Jinfen Zhang. In the dataset, we found cases with multiple annotations. For example, in the sentence below, the relation between NFD and MMP9 is annotated as both indirect downregulator and inhibitor. There are 430 such cases out of 17,288 cases in the training set. Because the ratio is small, we ignore this and still use single-label classifiers. We also found overlapping entity annotations. For example, in this sentence, two entities are annotated, GABA type A receptors and GABA. Based on the annotation rule, we let all of them have a false label. When processing the dataset, we mask entity names with two special tokens, chem name and gene name. For submissions 1 and 2, we used the BERT model. We took the pooled output of the CLS token, passed it through a dropout layer, then a linear layer, and a sigmoid function for class probabilities. For pre-trained weights, we found PubMed BERT trained on full text articles yielded best results. We also used a data augmentation technique. We first trained the BERT model, then found out all mispredicted sentences. For each of them, we removed the word not in the shortest path between chem name and gene name to construct new sentences. We believe these new sentences are similar to the original ones and can help the model on these mispredicted cases. For all models to follow, we all used this augmented training dataset to fine-tune. Here are the BERT results. For split 1, we used 80% training data as our training set, and the remaining 20% as the validation set. For split 2, we used the whole training data set to train, and the development set to validate. For the following models, we all used split 2. For submission 3, we used the T5 model, short for text-to-text transfer transformer. It is an encoder-decoder model based on transformers that for every text input outputs text as well. In our application, we treat the labels as texts when fine-tuning the model and use the Psi5 as our initial weight. Psi5 is trained on large biomedical corpora. See below for the performance of our fine-tuned T5 model. We also used the sentence BERT model to train embeddings of sentences. We used the batch-hard triplet loss when training the sentence BERT model. To calculate the loss for any selected sentence, we call it the anchor sentence. We first find a positive sentence that has the same label and a negative one that has a different label. Given any distance measured d and a margin m, the triplet loss is defined as follows. We want the distance between the anchor and the positive to be smaller than the one between the anchor and the negative by at least m. For the batch-hard triplet loss, we find the hardest positive and negative sentences within the mini-batch. Although not submitted because we are only allowed to submit 5 results, the sentence BERT model has 3 uses. First, use the k-nearest neighbor to train a classifier. Second, do clustering and help with model ensemble. 3. Sentence embeddings make more tools accessible for our analysis. We use the 2 types of ensemble methods. The majority vote algorithm takes the most voted label among BERT, T5, and the sentence BERT models. Whenever there isn't a majority, we take the prediction from the base model that gives the highest F1 score on the validation set. The trained ensemble method is based on the idea that individual model performance may differ in different clusters. We first cluster all sentences based on its sentence BERT embeddings, then train a classifier using the following features and response. For features, we use the cluster ID, class ID from sentence BERT, last layer BERT vector, and class probabilities from BERT. For the response, we use whether the models predict the labels right. T5 is not included because our time was limited. Here are the results for our ensemble methods. The majority vote algorithm yielded the best results in both the validation set and the test set. We believe the test result for the training ensemble method is bugged. Here we show the granular results by relation type obtained by the majority vote algorithm on a development set. We show two observations from our analysis. First, agonist activator, agonist inhibitor, and substrate product combined only have 67 of 17,288 instances. We think this is the reason that they have a 0 F1 score. Second, we analyzed the part of label that have relatively poor results. Thanks to the sentence word embeddings, we can find instances that none of its 10 closest neighbors that our models predict right. Using this technique, we are able to find several cases that we believe are mislabeled. An example is shown here. That's all I have. Hope you enjoy the rest of the conference.